מוכן, eh?
גם לקהל קטן אני מוכן.
Okay. So we'll continue our talk about the motor cortex. So the plan is to first lesson to mainly finish with the motor cortex. And then in the second lesson, I had a plan and I changed it. I want to do the, let's say, introductory talk towards the papers next week, which I'll talk a little bit about dynamical system, and I'll show an example, the neural integrator of a dynamical system and how it was studied in the brain. So there will be also independent content of what's going on, what will be in the papers, but also give you some introduction. So we were talking about what does the motor cortex do, and what does it encode, and what does it tell downstream areas. And we talked about Everett's experiment, where he showed that there's a lot of dynamic encoding in the motor cortex, encoding of the power that the muscle and we also talked about a different approach of Georgopoulos, that he talked about the direction of movement. He talked about the population vector, how you can take all you record many cells and take all the information from the motor cortex, or a lot of the information from the motor cortex, and get a very good estimation of the direction of movement. And we were talking, and here you can see again the experiment where a monkey moved in 8 different directions, and many cells were recorded at the same time. And you can see here each line here represents one cell, and the length of the line represents the firing rate, and the direction, the preferred direction of the cell. And then when you sum all this activity, you get this red or orange vector that represents very nicely the direction of movement. So as I said, up to now we talked about just an experiment where there's a very defined time of movement, and we're summing across a large time. You can actually also calculate the population vector for each time point, and this is the experiment by a group of Andy Schwartz. So what you will see here in blue, it's a little bit, can you see it? The blue lines represent the single cell. I think the white line here represents the actual arm position, and the yellow, I think, represents the population vector. And what you'll see the monkey doing here, this kind of spiral movement, and you'll see how nicely the population vector is actually estimating this direction. So here's the movement. Okay? So it's a little bit fast, and you can go and look at it afterwards at home. I'll put it on the web. So you can see that this is also the point here is that the population vector is also a very good estimation for a time-dependent measure of the direction of the hand. So we have all this information about the direction of movement. It doesn't mean that this is what the motor cortex is telling the brainstem or the spinal cord, but it means that we have that information. And as I said, we talked about the brain-machine interface. That was an example in a way in which you can use all this information to actually generate movements on computers or machines. But still, this is from in the 1980s. They came with the population vector, but still didn't answer the question: what is exactly encoded in the activity of motor neurons? The motor cortex? Is it related to the power, or is it related to the direction of movement? We still do not know. And yes, question? Oh, okay. Thought you raised your hand. Sorry. But still, we still don't know what it's encoding. So then I want to go over this experiment that was exactly towards trying to understand that. Are the cells in the motor cortex controlling the power, the muscles? Are they controlling the direction of the movement, or are they controlling perhaps maybe in intrinsic coordinates of the joints? So just for a few, it's hard to see, a few terms, maybe I'll put it higher. Can you see this? Okay. So just a few terms. We talked about extension and flexing, right? You should know that. Now there are two hand positions. Pronation is when you put your hand like this, and supination is the opposite. like this. And then there are two additional arm movements. There's radial deviation. That's when you move your arm like this, your wrist like this, and the other one is the ulnar, it goes like this. Okay, just a few terms that will be useful for the rest of this slide. So then, in this experiment, what they did, they had the monkeys holding a bar in two postures, either pronation or supination. That's what you see here. And then they made the monkeys move in 8 different directions. What do I mean by direction? So we have here a direction of so they're holding a bar and they're supposed to do an extension. That means that they'll push their arm up, right? And flexion will be in the other direction. That's 2 directions. And then they can also do forces like this direction, radial or ulnar, like this. So they have 4 directions, and they can also do a combination of the 2 muscles. So that's the movement the monkey is required to do, and he controls like that. He sees he gets, for example, visual feedback by a cursor on a screen, which he sees which will be proportional to the amount of power that is extracted. Okay, so that's a simple experiment. So they have now here 8 different directions: extension, flexion, ulnar, and radiation, and the combination of the 2. And they do the same thing in a supination. So the nice thing is that this differentiates between different coordinate systems of different coordinate system. So if you think about coordinates of the world, when the hand is like this and like this, and you want to move up, right? It's up in this direction. This condition is exactly opposite in terms of the posture of the wrist, right? So now up is like this. I'm doing a flexion in this case, and up like this is actually extension, right? So if the cells would encode in the coordinates of the world, you'd expect the cells in the motor cortex to respond the same if I'm moving up like this or like this, right? But if they're encoding in muscle coordinate, we might expect something a little bit different. So this is the movement coordinate. You can see that it's completely opposite, right? So what was extension is now flexion, and so on. And then we can think about muscle coordinates. Now this is not something that you can see by looking at the arm, but you can ask how much are the different muscles changing their preferred direction. So for example, this muscle, when I move towards the extension, where I move this muscle will be maximally activated in this direction. Okay, you see this? This other muscle will be maximally activated in this direction. Okay, and the other one will be in this direction. So we have your 3 muscles. This is in the pronation condition. In the supination, they change their preferred direction. And this is again an empirical fact. It's not something that you can see just by looking at your arm or I need to tell you about this, but they move around in 90 degrees. So if I look at this muscle, FCR, now this is its preferred direction when it's in pronation, and this is its preferred direction in supination. It's around 90 degrees difference. So the point is that if neurons will encode things in muscle coordinates, you'd expect a change in around 90 degrees in their preferred direction. Okay, is that clear? And then if it could be also in the joint coordinates. So this is coordinates that, let's say, that doesn't care about the direction in the world. It just depends on how much the relation between the arm and the joint. In this condition, if it's in joint coordinates, when I move down like this, it will be exactly opposite when I move down like this. Okay, in terms of the joint. So then, in this case, I would expect that the shift will be 100 degrees. And then the question, so they did this task, and the question, how will the cells in the motor cortex respond in this kind of task? So we have 3 different hypotheses: movement in motor coordinates, in world coordinates, movement, in this case, we expect no change in the preferred direction of cell, movement in muscle coordinates, which is expect around 90 degrees change, and movement in joint coordinates, which would expect 180 degrees in the change in direction. And here you can see 2 examples. So this is the rasters of the 8 different directions of movement and pronation and supination. Okay, and let's look first at this cell because it's very simple. So here this is in the world coordinates, so this is up and right, and so on and so forth, and you can see the neural activity in each of these conditions. And you can see that for this cell, it didn't matter if it's in pronation or supination. The response is very, very similar. You see this? So this means this cell is encoding the direction in world coordinates. In a sense, it's like Georgopoulos thought about things. But then we look at this kind of cell, and let's look at, for example, in here, up. So here, up, it has a very, very strong response, maybe the strongest up. This is in pronation. This is just extension, okay? Now, when we're in supination coordinates, then when we look at here, you see the response is smaller, okay? And if you look carefully, you see there's a shift here, which is around 90 degrees shift, okay? This is more or less. So the difference between, so if let's say this is the strongest response here, this direction, this will be the strongest response in the supination. So there is a shift in the preferred direction of the cell. And then when they looked at the whole population, this is what they found. So here you can see, this is a little bit complicated graph, but we'll go over it slowly. So this is the x-axis is the shift in the preferred direction, how much the cell shifted in the preferred direction between pronation and supination. The y-axis is the number of cells, so it's a histogram. Okay. There are two conditions here. There was one gray and one white, which are very, very similar. They depend on, do the cells generally are modulated by the posture, but we're ignoring that currently. And so this is for the cells, and this is for the muscles. And you can see maybe the muscles are around a little bit less than 90. So the muscles change their direction around 70 degrees. Okay? And so this is the muscle hypothesis. If all the cells would encode in motor coordinates, we'd expect them to move in this amount. This will be the shift in the preferred direction between the pronation and supination. And this is what they got in the cells. So they found many cells that didn't change their preferred direction. Okay? So it didn't matter for these cells if the hand is like this or like this. It always responded in a world coordinates or the direction of the movement. So these are these cells. And they claim they saw here a bimodal distribution, which we have some like this and others that are like this. These are cells that change in around 70 degrees. Like what are these cells? What are they responding like? What? Like the world, like the muscles? Exactly. Okay? Where's this? So these cells are responding like the muscles, as you can see here. So their conclusion was, and this is later there were more experiments that showed it's not that nice. It's not a really bimodal distribution as they claim, but that was their claim at least, that we have some cells in the motor cortex that have a world coordinate and others that are muscle coordinates. They didn't find any that are in the wrist coordinates. So they're all here or here. So their claim is that we have these two different populations. As I said, the other experiments show that it's more complicated. We have some like this and some like this. But clearly, in terms of what is encoded, we clearly have cells that are encoded in world coordinates and other cells that are encoding it, or it's a movement direction coordinates, and others that are encoding it muscle coordinates. So that was that. So we now know that there's information about direction of the movement. Many cells follow the direction of movement, but there's also information about the actual muscle activation. And there may be, I don't want to go into it, there may be a difference and this is again under debate, is there a difference in the location of the cells in the motor cortex? So some people claim there's slightly very different location. So maybe these cells are more related to the cells that actually project to the spinal cord, but it's still under debate. So that was so that was, I talked a lot about the coding of the cells in the motor cortex, but as we'll talk from now on, they don't encode just the movement itself. The cells in the motor cortex also prepare for movement. They also encode, for example, a selection between different targets. They're coding also a lot of higher order properties beyond just the direction of the movement and the actual command for moving the cell. So this is from the work of Georgopoulos, the same one with the population vector. And what he did here is a very simple experiment. The monkey is holding this arm, and then he has a cue, so he marked here with a red dot that tells him which direction he will go, he needs to go. And then after some time, there's a go cue, which allows him to go. So at the beginning of the trial, he's holding his hand in the middle. Then this dot appears, tells him that he will need to go in this direction. And then, let's say, the dot disappears, and then he needs to move in this direction. Okay, very simple. And so what this gives us is this allows the monkey to prepare for the movement. And what Georgopoulos showed here, that the population vector during the preparation will already tell you what's the direction that the monkey will move. So it tells us that we have all this information about the future direction of the movement, but it also tells us of a way in which we can decode it. Just take the population vector. And again, this is related to one of the papers that we will talk both of the papers that we'll talk about on Thursday are related to this. They at least claim this is not valid for, let's say, in a different context. So they claim there's no information in the population vector about the future movement, but here still Georgopoulos found it. So what do you see here? So time 0 is the time of the cue, when the red dot appeared, and then he has like around 500 milliseconds to prepare. And here you can see the population vector in time. You see this? Each line here represents the population vector, and the time is the x-axis. And let's say the monkey here is cued to move in this direction, towards up and the right. And you see there's some time when we don't see anything. So this is also very typical. There's some time until information gets to the motor cortex. But when it gets there, you see that the direction of the vector is like the direction of the future movement. Okay? So there is information about the planning in the motor cortex. And again, this is for going up. Simple, you see a nice up vector. And this is for left and right. Is this clear? Simple. Okay. And then Georgopoulos did something a little bit more complicated. He also showed that you have representation of mental rotations in the motor cortex. So the experiment is very similar to what you had before. So this is the experiment where A here is the experiment where he's cued to move to the left, and he actually needs to move to the left. But then there's another set of experiments where you put the dot on the, let's say, on the up, and he knows that he needs to move 90 degrees to that dot. Okay? So it's like this experiment. Imagine he sees this dot, and then he knows that if he sees a dot, he needs to actually to move in 90 degrees. Let's say he sees this red dot, he needs actually to move to 6. So what what the monkey needs to solve is basically a rotation. You see something, you need to do something that is rotated in 90 degrees to that. So first, the direct condition where it's cued and moves in the same direction, and this is exactly what we saw before. The population vector is directed towards the direction of the movement and the cue. But then when you look at the condition where you separate the two, so he's cued here in 90 degrees, and he moves in, he's cued up, and he needs to move to the left. And here you can see the population vector. So the x-axis now here is the size of the population vector, or the direction of the population vector. And the different lines show, this axis shows the time. So they do this population vector in time. And you see that in the beginning there's nothing, because it takes some time until the motor cortex information gets to the motor cortex. But then really, if you look carefully here, you see that in the beginning it's directed towards the signal, the cue. And then slowly it rotates, you see it, until it gets to the direction of future movement. Okay, so again, this is all before the movement, and this is the quantification of or this is showing this in a different way. So here, these are the first vectors, and you see how they move here from up to the left. And here, in the condition where it's direct, you see they're always to the straight line. Okay, so what they claim is that the signals in the motor cortex could also, let's say, they're not saying that it's implemented in the motor cortex, that they cannot prove with this, but they do say that they have the signal for rotating doing this mental rotation in space. And this is just the direction. So this is just the direction and time of the vector. So here it's already from the first time that it's big, because you can. Measuring angles of vectors when vectors are really small is very, very noisy. So they look only when the signal is big, and then you see that already when it's big, it's in this condition, it's to the left. In this condition, you see how it's rotating in time, starting from the up and then going to the left. So we have all this information in in the motor cortex, about planning the movement, about also mental rotation in the motor cortex. So you could think that you have two different populations, one that's coding the movement and a different population that actually is coding the direction. And it's very clear, and this is just an example for one cell, that the same cells that encode the movement are also the cells that do the preparation. And a big question is, okay, you have all this activity during preparation, why doesn't it cause movement? And that's one of the papers that we'll talk on Thursday. So you can see it. So here's the example to show that in the same neuron encode both. So you have very simple task, reaction time task. So it's again the same task where he moves the arm in a different direction. You tell him to move. He shows and then immediately when he sees this dot, it's already can start to move. Okay, target appears, and when the target appears, he already can do the movement. So there's no planning here beforehand in terms of direction of movement. At this point, he doesn't know the direction. And you see the responses here, and you can do a different, very similar task with preparation, similar to what I showed before. Again, he's looking at a dot, then let's say a white dot appears here, tells him where he should move, and then he has a go cue where he actually needs to move in this direction. And this is example of 3 cells, but generally when you look at motor cortexes, you see many, many cells that respond like this, that are not coding just the movement, but also encoding the preparatory activity. So this is the task, this is the reaction time task, this is activity, the example for activity in the reaction time task. So we have here some activity before, and then one point the target appears and he needs to move, and you see this large response during the movement. Okay. But then when you take the same cell and and it's now in the this task, the delay task, you see that there's a response to the visual cue. This is what you see here, and then it continues to respond during the task until the movement, and also during the movement it responds somewhat in this condition in this cell. So we have response both during the movement and during the preparatory activity, and you can see it also here. We have other cells. So now cells have very, very, very complicated temporal pattern, and when you go and record cells from the cortex, each cell will respond maybe slightly different, and it will be very, very complex. So here's another example that shows a different kind of pattern. So this pattern was just sustained activity. Here we have a pattern which responds during the movement and the reaction time, but when we separate it to the signal and cue, you see a response in the cue, right, and also the response during the movement. And you don't see much in the middle, maybe a little bit. And you have other cells. So this is another example. So this cell responds during the movement, right, during the reaction time task, also during the structure delay. But you can see there's this ramp in activity. You see this? And one important. So this is another common profile that you see cells ramping, increase in their activity towards the movement. And the question is, what does this mean? What does it mean that the activity is becoming bigger and bigger towards the movement? What is this related to? It can be shown that this ramp in activity, for example, is related to the time, to the latency of the movement. So how, if there's larger ramp, it will move faster or sooner. It'll be more prepared in a sense. There are other studies that show you can have ramp in activity that's related to the decision of the monkey. But this is a general pattern that you see in many, many cells in the cortex, not just in the motor cortex, also in other areas of this ramping of activity. But generally, what I wanted to say, the motor cortex is doing just more than just controlling more than just moving the arm. It also is related to the preparation and the planning of the movement. Here's another study that shows that. So this is a study from the group of Ronil Feromo, where they recorded activity in different areas: the motor cortex, the supplementary motor area. And you can see here the different and the sensory areas. So this is a task. So what the monkey is doing is putting his hand on some bar, and then there's a rotation. He feels that rotation, and then after I think a second or two, another rotation appears. And if the rotation is larger in the second, stimuli needs to move to the right, and if it's smaller in the second, it needs to move to the left. Okay.
Rotation of what?
It's like a vibration, not a vibration on the finger. It feels like a vibration with different frequencies. Okay? So that's what these two sinusoids represent. Vibration, and their density is the frequency. So in this case, so you have F1 and F2. You see that? And in this case, the second vibration is faster than the first vibration, so it needs to move, let's say, to the right. And if F2 would be smaller, then he would need to move to the left. Is that clear? That's a simple task. And when you think about it, it's a perception discrimination task, right?
Why does he have to look to the right or to the left?
That's arbitrary. If he makes the right decision, he gets reward. Okay. You can do also the opposite, right? So in a sense, it's a sensory discrimination task. You would think something like this would be solved if you would think of the classical hierarchical organization of the brain. You'd say, well, this is all sensory. Maybe the sensory cortex will figure this out, and the motor cortex will just tell you where to move it, how to move your hand. But that's not what they see. They see signals that are related to the decision also in the motor cortices. And I'll show that in a second. So here are 3 examples for C cells. So again, this is the time when the monkey feels the first vibration, and this is the time when it feels the second vibration. They do it with different frequencies. So here you can see the different frequencies. You can look at it later. The actual values are not really important. So this is F1 and this is F2. So yeah. So they organized it such that until here F2 is bigger than F1. So this is the cases when the monkey will make one decision. Let's say move to the right. And then you have the second set of trials which are symmetric, but F1 is larger than F2, and in these tasks the monkey will need to move to the other direction. So cells, you would think that cells that are related only to the final, to the motor act, will be divided by this, only here and only here. That's one point. And you would also think that the cells that are related to the decision will start to have interaction between what's the encoding at this point and this point. So here are 3 examples. So a sensory cell, on the other hand, if it's purely sensory, would not care about anything, would care only about the frequency, right? So here is this cell, for example, you see that it's responding mainly to the frequency. So let's see it here. So let's look at this trial. So at this point it's getting a frequency of 26, so the cell responds high, but afterwards the cell is responding in the F2, the frequency is larger and the cell has a larger response. Okay? When we look at the opposite condition, let's say this condition, okay? So this is 34 and 26. So first he's getting a fast vibration, and you see a large response, and also to the slightly slower vibration is also responding strongly, but less strongly. So this looks like a sensory cell that responds like the sensory input. Okay. But then we have other cells, cell like this, which it's hard to see exactly what is responding. But what you can see for this cell, that there's a lot of preparatory activity later. You see this? So in a sense, and this preparatory activity, this activity before the second F2, is basically telling this is how the monkey can memorize what is the first stimuli, right? He needs to memorize somehow to see what happened and to remember, because he needs to compare at this point because what happened before. And this activity will be, yes?
Is the firing rate, like the baseline of the second cells, are higher because of something?
So different cells have different baseline firing rate, and we don't know really what's the reason for different baseline. One thing that we do know is that inhibitor neurons tend to have a higher baseline, but they're also excitatory neurons with high baseline. We don't know. It's also many times task dependent, because if the cell is encoding something and you're controlling the baseline, and at the time of the delay before you actually start the task, it's related, what the monkey is doing or the animal is doing is related to its behavior, you'll have actual responses to that. But generally I would say we don't know what's the difference between the two. But this cell, if you can see it, so it's encoding, let's say, when the frequency is low, let's say the frequency now is F1 is 10. You look at the preparatory activity here, the delay activity is quite low, but when the first frequency is high, you see that it's high. See this? So it's encoding, it's encoding basically in time the frequency of the first stimuli. And then you have cells like this, which encode F1, and you can go over it at home, see the differences. But then at time F2 appears, this is what you see here. They're divided into two trials where the monkey responds very, very strongly to F2, that when F2 is larger than F1, then you have this large response, and then when F2 is smaller, you have basically no response. So this is a cell that's like the endpoint decision of the monkey. So these are just 3 examples. Well, they did some analysis. The details are not important. I'll tell you what the results are. Basically, what they do is now they do this linear regression between the firing rate and the different frequencies, and they get to their conclusion based of these coefficients. So they do this at each time point. They do this analysis. I'll show you. So cells in green are cells. So x-axis here is the time. Y-axis is the percentage of cells, and they have here 5 different populations. 2 populations in the sensory areas, that's what you see here, the top ones, and 3 populations in the motor areas, 1 in the premotor ventral, the supplementary motor area in M1. And basically, if you see here something green or red, it means that it's encoding the properties of the sensory stimuli. Okay? So in S1, you see cells responding in green here just because they're encoding the stimuli, the frequency of the stimuli. And then at the output, again, they're recording this stimuli, but not much beyond that. And also in S2, at the secondary sensory area, you see more or less the same thing. And so you don't see here any delayed activity as we saw before, like this cell. They're encoding, and basically this area is forgetting about this. But when then you look in motor areas, you see all kinds of complicated signals. So you see signals here in green. We said that it's encoding the sensory stimuli, but then it's continuing to encode it across all the delay. You see that? Meaning that this area contains the memory about what will happen. And then during S2, you have some cells encoding the sensory of S2. This is the red one. But you also have many, many cells that encode the decision or which direction, like this cell. The blue ones are like this cell, that are encoding which direction the monkey will decide to move. And they're also, it's called here interaction, more complicated cells, these black ones. But basically, you have here all the signals that you need in order to make the decision. And the point is that you have them here in this motor area, but you don't have them in the sensory area. So even when we think about sensory tasks that require sensory discrimination, the motor cortex and has a lot of information that's related to that. In this case, the supplementary motor area had also some of this activity, maybe less than PMV in this condition. And the motor cortex did not, and the M1 did not encode here the sensory properties or or didn't have the memory. It encoded mainly the decision in blue. That's what you see here. Okay. So this is Vernulfo's work is always really, really nice and really elegant. But this gives you already some kind of intuition, if you take it seriously, of how the brain makes these kind of decisions. So it gathers the information in the sensory areas, and then it projects to higher order, in this case, the motor cortices that are involved in using this information in order to make a decision. Some area that is missing here that is important is the parietal cortex, that also has a lot of information about the decision of the monkey. But it seems that what's happening is that the motor areas, or the higher motor areas and the parietal cortex together are coordinating the decision of the decision. And the point here is that this is not a decision, a high motor complicated decision. This is moving left or right. The hard part of it, in a sense, is discriminating the sensory information. But even in this kind of behavior, the motor cortices are the ones that were more involved than the sensory cortices. Yes. The black is interaction, complicated cells. Yeah, interaction between coding decision and encoding of the parameters of the stimuli.
Here, the y-axis just say, for example, for the green, the percentage of neurons that encode F1, like it's binary, encode F1, it doesn't encode F1, not how much it encodes.
No, just if it's encoded. How many are? I think it's how many that this regression was significant at each time point. So they take, what they do here is go look at each time point. They take a bin of, like, say, I think 100 or 200 milliseconds, count the spikes in the different conditions. So they have here these firing rates in a window, and then they do this regression analysis. I don't want to go into details about how the regression, but generally it's saying you would expect that if it's encoding completely linearly the frequency, you would get you get a cell that is significantly responsive to the frequency. Okay. There could be more complicated analysis to do, but this is a relatively simple and Nice one. Okay, so that's another example where motor cortex is related to more complicated functions like making a decision. And this is this is one slide. There's a whole field about how do we make decisions, how do we make sensory perceptual decision, deciding between two options. And there's a whole literature about whether the role of the motor cortex, the role of parietal cortex is a lot of debate. It's a really hot and important field, I think. Maybe last example for non-motor or non-directly motor growth of the cortex, of the motor cortices. Did you hear about mirror cells? So you'll hear about it again. Did you all hear about mirror cells? So this is the work of Rizzolatti, where he took a monkey and made it make a simple movement, grab some food, and during this task initially recorded in this area, this is V5, if you look at F5, sorry. F5, it's a motor cortex, and here around this area, okay? Recorded neural activity, and we knew already that neural activity in this area is related to the motor task, to the motor behavior. So he's grabbing this food, and you can just do the raster. So here's time. I'm not sure where he started grabbing at this, but these are repetitions. Again, action potentials, these 4 different trials, and you can see that before it's starting to move, we don't see much, and then when during the movement, you see this large response of the cell. So this is a classical cell in motor areas. But then he came with the idea to do this cool experiment, where he said, well, maybe this cell or this area is not involved only in doing this movement, but it's also involved when other animals or humans doing this behavior. So this condition, the monkey is just looking at a different monkey taking food. So we have here a monkey in a cage. I don't know. You bring the experiment brings a food to the monkey. This monkey grabs the food. This one is only looking at it, and again, this cell responds very, very strongly. Okay, so it's not responding to the arm movement itself. Also, when a different monkey is grabbing a food, it will also respond. And this is another condition where the experiment itself does take the food, so it's not species specific. Also, when humans take food, they also respond strongly to that. So it means it's more abstract. It's about someone else taking food. It's not a specific monkey or a specific hand taking the food. And this is the basic result, and later they did more experiments where they showed how specific it is. So there are cells that are responding when you're grabbing food in a specific direction. If you grab it like this or you grab it like this, and depends what you're exactly holding. You have cells that are specific to this, and these are the results. And then there's many interpretation what this means. So that this is the idea of a way in which a monkey can simulate what the other person or monkey is doing, right? Getting an idea of a theory of the mind, what the other person is doing. You do that by simulating it on your own brain. So now you're understanding. If you follow that, you have an idea what the other monkey is doing, and you can use that for many things, for social things, for example, and so on and so forth. But again, these are interpretations. At the simple level, you have your cells in the motor cortex that do, in addition to controlling the movement, they also are related to the control when other people are controlling the movement or other creatures. So that's another example. And importantly, so initially they did this experiment in F5, but now I think like 5 years ago there was a group that did the same experiment in M1, and they did not just in M1. They took cells in M1 that they identified them as cells that go to the spinal cord. So they did, I told you last time, you can find cells in the motor cortex that are in the pyramidal tract. So they identified those cells during the experiment, and then they looked, do these cells have mirror properties? And indeed, also in M1, also in the last station in the cortex, you see cells that are responding to they have these mirror properties. They respond when someone else is doing a movement. And then this again leads to the question, well, these are cells that are really close to the final motor behavior, and the monkey is not moving here, and you can do control where you show that there's no muscle activity. How is it that this kind of activity is not leading to movement? How is it that we have activity in M1 So you can think of it as and if we would do the same experiment and would now stimulate your the motor neurons, you would get a movement. So in that sense, these cells are not like that. They don't actually cause the movement, but still we know there are cells that are strongly related to the movement, and we see these modulation. How is it that these modulations don't lead to motor activity? And again, there are many ideas about this. One of the papers that we'll talk on Thursday will show an idea and some data analysis beyond it. How activity in the motor cortex, what are the properties of the activity in the motor neurons that will lead to movement, and other properties of activity in the motor cortex will not lead to movement. And this is again an important and a lot of debate and how that is happening. So you can think about one example that there's maybe some pattern in the activity, and that will be that paper, but you can also think of there's like a gate downstream. The spinal cord has this inhibition that when the monkey is preparing, it's all the time strong inhibiting the muscle, and then when you actually do the movement, you shut off that inhibition. That's another mechanism that could lead to this kind of you could lead to this dissociation between activity in the motor cortex and actual movement. So you can think about something downstream that is inhibiting the movement during the preparatory activity. In our movement, we don't know about that. In eye movements, for example, we know there's a population of neurons that are only when there's a movement, so they're all the time these are inhibitor neurons that have very high firing rate all the time, and then during the movement, just when the movement starts, they all shut down. These are paused, and they allow the movement. So you can think of that as a mechanism to stop a behavior when you don't want it to happen. Only when you release this inhibition, you can lead to behavior. So that's another example of how you can dissociate between preparatory activity and actual movement. So to summarize this part, the motor cortex part, before I'll go next lesson to a little bit about dynamical system, we talked about mostly anatomy of the motor cortex, input to the motor cortex from all different areas of subcortical and cortical areas. We talked about the output, about the descending pathways to the spinal cord. We talked about the spinal cord, the organization of the spinal cord. I gave maybe 2 examples for reflexes. We talked about the maps in the motor cortex, the relation between activity or stimulation in specific area in the motor cortex and what behavior it causes, what movement it causes. I talked quite a lot about the coding of single neurons. Is it force, or is it related to the direction of the movement in the world? That was the experiment of Everett, and the experiment that I talked today about of Kakai from 1999, in which they did the pronation and supination. We also talked about population, how you can extract information or ways to extract information from the motor cortex, and we talked about the population vector, which is a very good and which turns out to be very good in order to code the movement. Related to that, I talked about brain-machine interface, which is another way which we can use our ability to extract information from the motor cortex in order to move robotic devices, perhaps to help people with damage to the spinal cord. And then I said that today I talked mainly about, I'd say, non-direct motor properties of the motor cortex, about motor planning, decision-making, and mirror cells. I want to emphasize that maybe every behavior that we do has a motor component. Okay, even the simplest, even if you you sit in the class and you write in your notebook, you think you're studying some abstract way, but there are motor implications to whatever we do, and it's not surprising that almost everything is related in some way to the motor cortex, because everything has, and to the motor part, because everything has a motor component, and it turns out it's not just the motor component. You can think about more higher and more complicated functions that the motor cortex does. Questions before we go to the next part? You're all ready for the test? Okay, so maybe let's take a break, and then so I'll talk about the dynamical systems. I understand some of you learned about differential equations. Some of you did not. I will mention the details. I also give the intuition for the thing. So if you don't know how to solve a differential equation, it's okay. A linear differential equation, it's okay. If you do know, it will give you maybe some ideas of how you can use it in the context of motor control. And again, this is also introduction for the papers, and also I'll show you another example of how the brain, a computation that we know the brain implements, how it could implement it. That's the Neural Integrator. That's 10 o'clock.
זה נורא.
There's like some kind of rotation. Just like different kinds of movement. Yeah, Bon, mais enfin, bon, honnêtement, okay, you're better. I feel like yeah. You ever been to Seattle?
Never. But I can say that out of you.
Right, that's what everyone knows it for.
אני אזכיר אין שום בעיה. כן, אז אני אזכיר את זה, אני לא... תראה, אני אזכיר את זה ואני אביא דווקא דוגמה אחרת מה שאתם, זה יהיה הרקע. אני לא אדבר על מה עכשיו לדבר, אני אדבר על Dynamical System אחר, שזה ה-Neural Integrator. בסדר? ביחד נצליח אולי להביא משהו.
אף אחד לא פה, זה בקטע. כן.
קצת סובך, אבל אל תיבא לי.
זה באופן אינטואיטיבי.
אני אנסה להסביר גם באופן אינטואיטיבי. זה בכל זאת קורס באלסק, אז חשבתי שאפשר.
לא, אבל באמת יש האינטואיטיבי 5 שלו. 5, 9, כן, 5 שלו. אולי 5 שלו גם...
מה זה?
אולי בין 5 שלו אולי גם הם כן יודעים, אני יודעת. חוץ ממני. ביקשתי מהבן שלי לעזור לי להתאחר מה יש לחדווה בכלל.
כן, לא, זה חדווה מסובך קצת אפילו. אפילו לא חדווה אחד.
לא, למדתי לפחות 2 קורסים בחדווה, אבל אני כל כך הרבה שנים שהם לא זוכרים כלום. בקושי אני זוכרת את העיקרון.
יהיה גם נתונים רגיל.
כן. אני לא בחרתי, קבעו לי, אני זוכר 3 קורסים לעשות, לפני שאני יכולה להתחיל לעשות את הקורסים של הדוקטורט עצמו, זה סתם קורסים בלי...
כן, טוב.
רק ממוצע אני צריכה. שזה לא מעט.
כאילו מחר, אני לא התחלת לעשות בעסק. לא התחלת לעשות את קורסים בעסק.
אני לא קשורה לעסק.
יש קורסים מקבילים לזה שהם בפקולטה.
אני שייכת למדעי
ושנת תואר של תואר מתקדם.
מדעי מוח, מדעי המוח והתנהגות ומדעי החיים.
הבנתי. יש להם קורסים של קורסי בסיס שלהם.
כן.
שהם קצת יותר פשוטים מבחינה חישובית.
כן, כן, ברור. לא, קבעו לי.
קבעו לך? לא הייתי יכולה לזה?
לא יכולתי לבחור, וגם לא מספיק יודעת כדי לבחור. הם רצו שאני אבין את המוח אם אני כבר בתוך החוג שלהם. הסכמתי איתם. אבל בחרו לי. המנחים שלי, יש לי 2 בכל זאת, ולא...
מי ומונה ו?
ארי מרסון, אתה מכיר? אבא שלו הוא פיזיקאי פה, הוא פרופסור פיזיקה פה. הוא מנחה שלי, אנחנו לא קשורים.
Okay, so this is the experiment. Can I teach dynamical system, linear dynamical system in an hour? I'm not expecting you to understand all the math very deeply at the end of this, but I do want you have the intuition of what we can do with a dynamical linear system. So in this talk, I'll present the dynamical system and some solution of a linear dynamical system. And then on Thursday, Musa and Amir will talk about in how modern experiments use these dynamical systems in order to understand what the motor cortex is doing. That will be Thursday. Today I'll talk a different implication of dynamical system, I think that's called the Neural Integrator. So this is a network that was studied, was recognized its need in the 70s by a person named David Robinson in the context of eye movements. So it's called the oculomotor neural integrator. Later, there's also, we talked before, the lesson before I talked about these ramps of activity. So there's another field that talk about the need of integrator for making decisions. So that is the neural integrator. I'll talk about what kind of dynamical system will lead to these kind of functions. Okay, so what is I wrote introduction to dynamical system. I would say a reminder of dynamical system would be better. And if you don't remember it, then if you never learned it, then again, as I said, it's not the math is not critical, but the intuition I hope you'll get. Okay, so what is a dynamical system? We have some kind of variable x. It could be a vector that changes in time. We know something about its derivative. It's related, the derivative of x, or the direction in which it's moving, depends on some kind of complicated function on x, plus some kind of input, okay, to the system. This could be as complex as you can imagine, and generally it's hard to really understand what's happening here. We can look at the linear dynamical system, a much simpler problem, okay? So now instead of f, we have here m, so m is a matrix. So we have here a vector with different components that change it. We ask how it's changing in time, or the derivative of it, and that equals some matrix times x plus some kind of input, okay? So that's a linear dynamical system. And in linear dynamical system, we can do all kinds of interesting stuff, and we can get interesting solution and interesting problems for the system. And we can also have some analogy to neural networks. So just generally, you can think about this m. You can think of x of many, many neurons, for example. We're now talking about, this is a very, very crude way of looking at neurons. You can think of this as a rate of a neuron, the fine rate of the neuron, something that doesn't really exist in some sense. But still, that's how we think of it. We can think about this m as a connectivity matrix, the connectivity between different neurons, and x again is this is the derivative of the fine rate, and this is the actual fine rate, and this is the input to the network. This is the intuition of how we think of this as neurons. I'll show an example with more details in a second. So what are the solution of this system? Who never saw this kind of figure? Okay. So this is the solution for when you have two components, an x and a y, or x1 and x2, okay, where the derivative of the x the x depends on its derivative of x depends on x, okay. And the question, what will this system do in time? And these are the different solutions that you have. I'm not going into all of them. I just want to mention some. So each of these lines represents a trajectory in time. This is a solution, okay. So, and the solution here depends on these parameters, a, b, c, and d. Okay? Different parameters will give us different solutions, different behaviors of the system. So let's say if we're in this regime, so you see this parabola? All what's above it. In this regime, we get oscillations. Okay? So for example, let's say we started, this is the dynamical system, and we started in this position. What will happen in time? X1 and x2, or x and y, will evolve such that it will move in this direction. Okay? This spiral outward. And we have different solutions. Here it's spiraling inward. Okay? And we have solution where it's a constant circle, like these oscillations. You see it? So these are all solutions for the dynamical system. What's common to all these solutions? That they spiral, and the reason for that is that in this region, the solution, there are complex numbers involved in the solution. In a second I'll talk what is the solution. These complex numbers, when you solve it, lead to, if you remember, cosine and sinus is in the solution. So basically, these oscillations are cosine and sinuses evolving in time on the x and the y axis. So you have different kind of solutions. So for example, if you're here in this condition, this is a very typical condition. If you start in any point, you'll move outward. You see this? Here you're moving outward. Here you're moving outward. Let's say you're here, then you'll continue to move in this direction. So x and y will get larger and larger, in this case exponentially. Okay? We can be in this area. If we start here, then they all converge. X and y will get smaller until they get to this point. Okay? So these are the, let's say, there are all kinds of different kinds of behaviors. In one axis, x could grow, and the other axis it could shrink. So that's, I think, this condition. And we have different kinds of solution. The general solution for simple problems looks like this. Okay? So we have a equation that looks like this. X is function of x, of a matrix, and now x is a vector and m is a matrix. The general solution, when m has good properties that you can diagonalize it, looks like this. Looks complicated, but actually it's very simple. So xf is your starting point. This is the eigenvector, the left eigenvector of the matrix M. This is the right eigenvector. These are all, you can think of all these as in a sense as constants. In some sense, what's important is this part. So how will x behave in time? It will be an exponent with this is the eigenvalue times t. You see this? And this could be in many, many different directions. So if we think about this, let's say all these are real. if all of the eigenvalues are real, then this simply represents exponential growth in different dimensions, like you have here. Okay, in one dimension it's going to the, let's say, out, or exponentially shrinking. Or in the other direction, it's going here. It's going in both directions. It's going out. This is another example with all real values. But then these could also be complex numbers. And when they're complex numbers, you can organize this function such that it's not that it's not a solution in the complex. It's like Fourier transform. It's not a solution in the complex plane. This is a solution in the real plane, where you can organize the equation such that in each axis it will be an exponent and sinusoids or cosinuses. So it will look like this. Okay? These will be the solutions. This is in 2 dimensions. So the way it happens, these r's and lambdas are such that they come in pairs, conjunction pairs, where you can change just organize the function such that you'll get planes where you have these kind of rotations in the real plane. Okay? And generally what we had here in this part were conditions where these values, these eigenvalues, were imaginary, or had an imaginary component. Okay? And in this condition where it's only moving like this, and it will become important later, these are conditions where it's only imaginary. There's no real value for the function, for this equation. Okay, so how can we use this? So this is now you know dynamical system. We can continue. Let's think about one example, a simple example for this. And this is where physiology. So I want to show how we can use this for actually studying physiological things and real things in the brain. So here's a very, very simple example where I have a dynamical system. In this case, it's still one-dimensional. Okay, it's called the Neural Integrator. In a second, you'll understand why. So we have here one neuron, and you look at the derivative of the firing rate in time, and it equals some kind of, it's related to the R, to the firing rate, plus some kind of input. Okay? And just to make it, you have here some kind of time constant of the neuron. Again, this is mathematically, this you can just divide by this, and it all will be like what you had before. But in terms of physiology, this represents like how fast the neuron will decay to baseline firing rate. So imagine we don't have this WR. Okay, we have something R. A derivative equals to minus R. Imagine that the W there doesn't exist. So you have some derivative equals to minus itself. What will be the solution for that? Exponent, right? It will be a decaying exponent. So in these neurons, if you just do nothing, if you don't have this connectivity, the neuron will just decay back to 0. Okay? So that would be the solution. But then we have this W. And now, in this condition, when it's a single neuron, imagine W is equal 1, right? Then you have minus R plus R. This will cancel. And then you have if this is the condition, then you have dr dt equals I. And what is the solution for that? If all this is 0, this is what you see here. Exactly. It will be the integral of the input. You see? So this is the condition. So we started with this simple scheme. We went to this. This is the condition when W is equal 1. And then we have that the derivative of R in time is equal to the input of the system. And then you have R is just the integral of the input in time. So this is why we call it a neural integrator. It integrates the input that the system got in time. In a second, we'll see an example why it's really important, why all of you have an integrator in your brain. So you can implement an integrator with a single neuron. So you can imagine you have your neuron, and it has now an auto synapse on itself. This is what you see here. And now if this W, so the way to think about it, this minus R is just for the neuron to decay in time to 0 if there's no input. And this W is what will allow the neuron to keep its firing rate high. And if this W is equal exactly 1, then you get here that the rate is constant. The rate does not change in time. So you set the neuron to some kind of rate, and the cell will stay in that rate. And this is the firing rate of the cell and how it will change in time, or what will happen to the neuron, to the network if you're at each point. It will stay in the same point. And the analogy or the intuition here is that you have here the network is in a state such that it's on this plane where you have a square on a plane. In any point that you will put it, it won't change its location. And let's think about this simple network with one neuron at this condition. So we have here now inputs to the network. You get an input. The input is integrated, and then the network becomes flat. You see this? Stays constant, and then another input. It integrates this input, and then the network gets to the next state. And then you integrate it. Another input comes here, and then it gets to the next step. Okay? Does this remind you anything? These kind of graphs? What do you think will look like this? So later we'll show that the eye movements exactly look something like this. So the input will be these brief inputs, and then the eye will look like this, or the firing rate of the neurons that drive the eye will look like this. And then if W is not exactly tuned, for example, let's say W is too small, smaller than 1, okay? And then the solution of the network is of this slide in time, okay? Just a decaying exponent. And that's what you see here. Now if W is a bit too large, then the system will just grow very, very fastly exponentially. Okay? So each time it gets an input, you give it a small input, it starts to grow exponentially. You give it another input, it just continues to grow exponentially, and so on and so forth. Okay? So so this condition is what we call a neural integrator. This condition, if the cell is tuned exactly like this, then the network will integrate. As I said, this is an analogy of one cell. One cell does not integrate by itself, and there are studies that show that. So you can go to a cell, disconnect it from everything, from other parts of the network, and you'll see that by itself it does not integrate. So we need neural networks to integrate, so we can generalize this idea of neural integrators to the same equation that we did before with a network. So this is just the equation that I showed before with many neurons. Do you see that? Did I lose you all? No? Yes? So this is just the same equation of the neural integrator. This is like this equation exactly. Just now we have many neurons. So you can think of each neuron each x is a neuron, and we have the derivative of the neuron. It equals to the minus itself. We have this term for the neuron to decay in time. Okay, so if this would be the only thing, if we would not have all these terms after this x, the neuron would just decay exponentially in time. But we have these inputs, and now we can really think of them as inputs. So these are inputs W, I, J. These are inputs from neuron J to neuron I. Okay. And you take the activity in neuron xj, you multiply it by wxj, and that will be how the system will change in time. So this is now a neural network, and it has some input. So here I wrote it U, but I is also. Here I changed it to U because it'll have I for a different symbol. But this is the input to the network, an external input. Okay? So now this is not different than what I showed before. This is still general. It's a linear general dynamical system. I'm just rewriting it, okay, in a matrix. So this is now for each component, we can write it as matrices, okay? So X now is a vector. This is a number. These are now two matrices, okay? W is a matrix of connectivity. I is the identity matrix. This is why I did not call this I, okay? And if you remember before, right, we in the beginning, we started with this M. You remember that? So now this M is exactly, this is just changing the names. It's just to make it like more physiologically relevant, right? So you can think of this M, if I just take this and divide by the time constant, you get the M from before. It's exactly the same thing. So now we have, this is a matrix, and we can have, and this is again the activity of the neurons, and we have some input. And the question, before we showed how this kind of network will integrate, we can also have this kind, of course, if one cell can integrate, also a network can integrate. And the conditions are a little bit different. So now, in order for this network to integrate, okay, you need to look at the eigenvalues of W in this condition. So if the eigenvalue, the leading eigenvalue is 1 of W, and all others are smaller than 1, then the network will integrate. Okay? We'll do exact integration. The intuition here, remember, the intuition is exactly what we had before. Before we had W, now is a matrix, right? And before we had W, we had a W that was a number. So in some sense, this W, if it has an eigenvalue of 1, it's like taking -1 plus 1, and you get rid of this component. Okay? The more mathematical, those who know math, the more mathematical, robust way to do it is to use, you can diagonalize the matrix, and you can change the name of the parameters x using this diagonalization to get very simple equation, which is solved and show that it's an integrator in some dimension. Do you want me to show that? Then let's do that. How much time do we have? So let's. Do you see if I write here? So we have x dot, not like this. So we have x dot equals m x plus mu, and now these are all matrices. Now m is diagonalized, right? We can write it as u minus 1a u will equal to m, right? We can write that, assuming that we can diagonalize it. Sometimes you can't. Now if we write a y that equals ux, okay? We can write this, change this the name. This is the linear transformation using this same u, okay? And then we can just plug this into the equation, and what we get, so this is, right, this is very simple. This means that x, y minus 1 of y, right? Now in each position where we have x, we can write this. So we can have u minus 1 y dot equals, now we can write instead of m this, u minus 1 a u, right? And now instead of x, we write u minus 1 and y. I had two u's. Let's change this to I. Plus I. Okay? Now this is identity, right? You see this? This is identity. Now we can multiply the whole matrix, all this equation by u on the other side, right? So we get, here I multiply this by u times u minus 1, right? I'll get y dot, I think. And then here I'm multiplying by u on this side, so I have u minus 1, right? This is I. A is left. This is also I. So what's left here is A, y plus, and I multiply u, I. Okay, this is the equation. Now this is the diagonalized equation, right? This is A is now a diagonalized. You see it? UI. Yeah. So A is right. A is diagonalized. That means that I can write this as many different equations, right? What's written, basically what's written here, I can write it as UI equal UI dot equals the leading eigenvalue u plus something that is not related to y. That's u's and t's, right? And this is a very simple equation, right? So you can solve this with the exponents and the way you have, and then go back and get the solution for it. So that was short nap of it. So we have here, so I want to go back to the linear neural integrator. So as I said, in this condition, right, so in that dimension, what's left here, and this is the difference between n and double minus 1, in that direction we have here basically a 0. This becomes 0, and then we have y dot equals to something that's related to the input, right, like we had before. So that's the neural integrator. So the condition for neural integrator is that this eigenvalue is 1 and all the others are small. That will make it an integrator. And we can have other solutions. That's what Musa will talk about, the intrinsic oscillations. You can think about other solutions. So let's say we can split any matrix M here to two kinds of matrices. And if this part is 0, and this is a symmetric matrix, and this is a skew symmetric, look on the internet what that means. But if this is 0, that means that all the eigenvalues will be imaginary. This is and then this condition
the network will just oscillate. And that's equivalent to this condition. Okay? And that will be important for the talk on Thursday. Okay, so we have now a neural integrator. How can we study the neural integrator? So I want to show an example. What do I mean by neural integrator? And where it started, and example for work that I did where we try to understand in the framework of neural integrator what is the meaning of the dynamic of the cells. And neural integrator is interesting because it's example. It's rare that we have example that we know what a system is doing. If we know that the system is in some sense or close to a neural integrator, it's something that in neuroscience generally doesn't happen a lot, especially in motor areas. We don't know what is the transformation that an area is doing. We don't know mathematically how to characterize it very well. And you saw that in our talk. We don't even know what's represented in these areas. And a neural integrator is an example where we have the mathematical background. We have some idea why the network, we think the network is integrating. So it's a nice way which we can connect theory and actual physiology. So the kind of neural integrator I'm talking about is the oculomotor neural integrator. This is the neural integrator that allows you to keep your eyes stable in time. Okay? So what do we see here? This is the eye movement. At one point, you move your eyes from one place to another place. This is called a saccade. These are the fast eye movements that we do in order to move our eyes from place to place. Okay? And then the eye stays stable in the new location. You see that? So you moved your eyes to the side, and it stayed on the side. And then you made another saccade, and the eye returned, moved back to a different location. What is important that we move our eyes, and then it stays stable. When we look at the signal that's actually driving this movement to the brainstem, or the input to the brainstem, we find these kind of cells. So many cells in the cortex, also in the superior colliculus, if you ever heard about the structure, have this kind of properties. They have this burst in activity that's happening just at the time that the eye starts to move. Okay? So this explains how the eye moves from one place to the other place, right? But it does not explain why does the eye stay stable. And in a second I'll show you what happens if there would not be any more activity beyond this, the eye would drift back to the center. Okay? So this is the activity you can think of. Most of the brain, most of the cortical areas have this kind of activity, also some subcortical area. But then this is the kind of the input the brainstem gets for the eye. But then when you look at the activity of the brainstem, you start to see cells that responding like this. Okay? So you can think of this as the input, and this is the activity of the cells in the brainstem. So you start to see cells that get this kind of input, but then they have this sustained activity in time. Okay? And you also hear some are completely sustained. Some have also respond during the saccade, during this movement, but also have some sign sustained activity later. And when you look at motor neurons, you get this kind of activity. Okay? You see this burst in activity and then this sustained activity. And this burst allows the eye to move from this position to this position, and this sustained activity allows the eye to keep stable afterwards. Okay? So what we have here, this, I talked here in a few minutes about activity in this cell, but what we also understand here the transformation, right? We have this burst in activity that needs to cause this sustained activity. And this is exactly what I showed, the kind of things that we want from an integrator before, right? We have this kind of inputs, and we want to integrate them in order to get a sustained activity. So this was recognized by a person that's called David Robinson in the 1970s. And then he proved that we need a neural integrator, and it's not just for these kind of movements. All different kind of eye movements need neural integrators, and he also located them in the brainstem. So what he did here you can see normal eye movement of, I think, a monkey in this condition. So he's moving his eyes. He's put the position. So x-axis is time, y-axis is the position of the eye or the angle of the eye, and they move from place to place, and they're very stable. And then he started to lesion different areas in the brainstem, around the vestibular nucleus, and what he found is that this is now the behavior after the lesion. So the monkey was able to do a saccade, you see it, but then the eye drifts back. And the idea here is that you have this kind of burst of activity, but you don't have the sustained activity afterwards. And this sustained activity you need in order, so the eye, you can think of the eye as a spring. So in a sense, when the eye moves to one side, you need some power against the spring in order to keep it stable. If you don't have that power, the eye will drift back to the center, and that's what you found here. So that's why we know we need a neural integrator, and we can ask what kind of activity do we expect to see in the neural integrator, and what will it tell us about the neural integrator? Again, this is in the context of dynamical system. We know that neural integrator is an example for a dynamical system, right? And we can think what are the signals that emerge from this system, and how does it constrain how the neural integrator is implemented in the brain? So, that was the example for neural integrator during saccade. We also need neural integrator for smooth pursuit. So smooth pursuit, I think I talked about. These are the movements that we do when we move our hand, like this, from side to side. We also need a neural integrator for pursuit, and here's the proof for that. So this is the system, and you'll talk about cerebellum and the brain. So in this system, there's the cerebellum projects to the brainstem that drive the motor neurons, and then you get this kind of eye movement, okay? So you see this. Unlike the movement before that was 40 milliseconds, this movement is longer, around 600 milliseconds in this case. It's just tracking the target that is moving. So that's what you see here, a very slow movement. And when we think about what are the inputs to a brainstem, we can have an estimation of that. And this is, I'm not proving this, but this is an estimation of what is the input to the brainstem. And we can also look at the output of the brainstem, and it will look like this. So this is the activity in the motor neurons. And our evidence that the network is integrating is that if we take this activity and we just do the integration, the mathematical integration of this, we'll get something that's very similar to this. So I'll show you how this looks. So what you'll see here is how this evolves in time. And then beneath here, you'll see the sum of this activity, which is basically the integration here. So you see this is how it evolves in time, and here you can see the integration of it. So the red line here is the area under the curve, right, under this function, which is essentially the integration, and the red line here is just this number, just summing this activity, and the blue line is the actual activity of the motor neurons. So we have here the input here to the brainstem. We looked at the input, and we looked at the output, and we found, by this simple simulation, we know more or less what the transformation is. So the transformation is equivalent to a neural integrator. I'm saying that if that would be the only evidence, then I would be concerned. We also know from other systems and from the saccade that I showed before, that that's what the brainstem is doing, and from the lesions. So the question is which kind of dynamical system will explain what we see in the brainstem? Or how does the brainstem do this kind of integration? And that was our question. Again, in the context of dynamical systems. Is this too fast? Are you following, or everybody awake? So you need to do experiments, and you want to record it. So the idea is now let's record in the brainstem, and we'll see what are the kind of patterns that we see in the brainstem, and what do they tell us about how the brain does the neural integration. So to do that, you need to record in the brainstem. Let's skip this. We record in different populations in the brainstem, extracellularly, and we get this kind of response. I don't want to go into the details of the recording; a bit complicated, and we don't have much time. But this is important. So we recorded 3 different populations in the brainstem. These are closest to the input. That's why I call them input layer. We had some way to identify them. These are cells that are not close to the input. They are further away. They're not connected to the input. That's why I call them intermediate. And this is the output of the system. These are the motor neurons. Okay? And these are the kind of patterns of activity that we found. These are average activity. And what do we see here? So I'm reminding you, the input to the network is something that looks like this black line. You see it? This is the input to the network. And this is the activity that we actually record. So in this, the cell that get the input from the cerebellum, we see a pattern that's very similar to the input, or not exactly like the input, but similar to the input. We see this transient response, and then some sustained activity later, and then this dip in the end. You see this? Then we look in intermediate levels. We don't see this transient response anymore. We see something that's mainly sustained. In the output, we see this rampant activity. Okay? So again, what are we doing here? We know this is a network that's integrating. We're going to this network, and we're probing the activity in this network, and we're asking, what are the patterns of activity that we see there? And we see this, we see this gradual transformation. We see in the input layer, the first cells in some sense have this transient, then the network afterwards does not have this transient, have mainly sustained activity, and then the output has this rampant activity. So what does this tell us about integration? That's the question. We are recording now in the Neural Integrator, and we want to tell what kind of integration methods, what kind of dynamical system will lead to these kind of patterns. So we can also first look at single cells. So this was the average of the population. And the single cell, so now each line is a single one neuron. Okay? You see this is the red are the same red from before. The cells that are closest to the input. We have some intermediate and then the output. And here we saw this transit in many, many cells. This transit response in the beginning. Here we saw mainly a lot of variability in this level. So these are cells deep in the network that does the Neural Integrator. And these are the output of the Neural Integrator. Okay? So again, this is a system now that cells are all connected to each other. In that sense, they're a dynamical system. But they also get input, and we know what they do. They are a Neural Integrator. They take the inputs that we showed before and then integrate it and bring to this kind of behavior and this kind of activity. So what kind of models, the question we wanted to ask, what kind of models will lead to this kind of patterns? I don't want to go into all the details of the system and how we model different populations, but what's important is that we can now, what we're changing, what we're testing is different matrices of connectivity. And we can ask which of them will lead to the patterns that we see, which of them are dynamical systems that lead to the kind of pattern that we see in the cells that we recorded. So we can think about many, many different connectivities. We can think of this. So this is just the anatomy of the system. Not really important. Just important that there is a cerebellar input, okay? And then it goes to the brainstem. So whatever here is the red is the brainstem, and we record it from this activity, okay? And then we ask what will happen, what kind of connectivity. We can assume just everything is random here. All the connections are random. This could be an assumption, and we can ask what kind of patterns of activity will emerge from this dynamical system. That's one possibility. Generally, random networks will not be neural integrators, right? We had a very specific pattern for that. But what we can do so if you remember here, we have this. So we can make we can generate a random function, a random connectivity, and then divide the whole matrix by a number to make it a neural integrator, divided by the leading eigenvalue, and that will make the network integrator. So it's random with another constraint to make it an integrator. We can think about different kinds of connectivities. So this is a connectivity where cells are strongly connected to the next level, but get only weak feedback. Okay, so there is enough feedback in this system for making it an integrator, but the important thing is that there is a strong feed forward. And this model was shown to be true for the neural integrator of fish, or similar to the neural integrator of fish. And we wanted to test, will it also fit what we see in monkeys? Maybe to make a link between different kinds of patterns. It also captured our intuition. If you remember, we saw this gradual shift from these kind of cells where we saw this transit response to these cells that have this sustained activity. So we want to ask, well, is this model fit what we see? And then there was other models where we asked, is this symmetry? We made it a strong feed-forward connection. We want to ask also, are these, if we make them symmetric, will it do what we see? And again, the question is, so these are different dynamical systems. What defines the dynamical system is this M matrix. So these are the actual M matrices that we use. Okay? These are matrices of connectivity between cells. This is the heart of the dynamical system. And these are matrices that make them integrate, but they will be different because they will cause different kinds of patterns of activity. So when we look at this brainstem as a dynamical system, we want to ask which kind of patterns of activity will emerge from these kind of connectivities. So now this is the data that I showed before. And now we can run these models, very simple models. I'm sure each of you can figure out how to program them by yourself. Not very complicated. And we can get this kind of activity. So now this is a model, right? This is the result of a system where I know what the input is, right? I gave it the input that I estimated from the cerebellum. I know what the output is. The output is the integrator. And these are the patterns that emerge from it. Okay? These are the different patterns of activity. And the point is that this is a very, very naive model, a very simple way to look at things. But still, we captured a lot of the dynamics that we see in the single cells. So, for example, in the single cells, we saw this transient, and we also saw it in our activity, in the activity in the model. Afterwards, we see this in the red ones. We see the sustained activity. Here, we see also it. We see less less diversity here, but still there is some diversity. Again, this is very naive. All the inputs to all the cells were exactly the same. You can imagine in real life the inputs do not need to be exactly the same, so you can get more variability in the actual data. In the end, we see this dip in activity, for example, in these red cells, and we also see it in the model. Okay? And also the green cells had a lot of diversity in our data and also in the model. The details are not really critical. What I want you to understand that we took here a dynamical system approach. We asked, we know this is a dynamical system. What kind of models, and let's try to fit, in a sense, our data to a dynamical system. Which dynamical system would be the best? And this dynamical system is good. We can ask, we had some other hypotheses. So a random network like this, where everything is random, you would not expect this kind of diversity. All the cells will respond the same, like the output of the integrator. And even the symmetric connectivity did not show all the properties that we saw before. So let's reiterate what I did. I know it was a little bit fast, but I'll try to understand. I'll try to summarize it. So we took this approach of dynamical system, which we know the cells are somehow connected. We said this is the kind of connectivity that we expect in this network, or this network is a dynamical system. We can have, some cases, in the case of a neural integrator, we know what the system is doing. It's doing an integration. We went and probed the actual activity in this network by recording the neural activity, and we asked what kind of temporal patterns will emerge from this system, and what did they tell us about the dynamical system. So that was in the context of neural integrator. Musa will talk, Amir will talk about a different kind of dynamical system, which will be the motor cortex. How can we fit the motor cortex for a dynamical system? It won't be a neural integrator. It will be a different dynamical system that what it does oscillation in the world. And that's the idea of that paper. Another idea, how it's still related, I want to return to the motor cortex. It's still a work about, it's still a course, this part of the course is about motor cortex. So it turns out neural integrators are also relevant for the motor cortex. So here you can see the activity of one cortical neuron, while this is a monkey. You record cortical activity, and you can also record the spinal cord activity. So this is from the lab of Ifat Prut. I don't think you made her because she's in a sabbatical, maybe next year. PI in LSEC. So she recorded in the same task I talked before of Kakai with 8 directions, pronation and supination. She recorded the activity in cortical neurons and the spinal cord neurons. Okay, and these are interneurons of the spinal cord. And you can see here the force that the monkey is doing. So he's moving his arm, and at one point, time 0, I think it's the go signal, or time 0 is the time of the movement. And then there's a power that, you see the force here, and then he needs to sustain the force for some time, for around a second. Okay? And then he can release his hand and return to rest. And when she recorded cortical activity, she found many cells that respond like this cell, okay, just at the time of the movement, when the movement starts. But then when there's a sustained force, was not much activity, okay? But then when she looked at spinal cord cells, as you would expect, because in the end, the spinal cord needs to produce the force, and these are interneurons, not motor neurons, but still you see this, in the spinal cord, you see this sustained activity, okay? As we saw before, you remember in the saccade, we were talking about sustained input for a transient input for a saccade that leads to sustained activity. Also here we have transient input to the spinal cord that leads to sustained activity. And generally this was true in this kind of task at least. So this is different than the task that I showed before in the sense that the monkey makes a force and then he needs to keep his arm for a long time. So unlike, for example, the Georgopoulos where the arm was moving all the time. And then they looked, so this is one cell, when they looked at the whole population, so this is time 0, I think is again the time of the beginning of the movement, some kind of normalization of the activity, so it will be in the same scale. And so the red cells are the cortical neurons, they increase their firing rate and then they decrease, although the EMG is still high here. So this is the cortical, the blue cells are the spinal neurons, you see them? They go up and they stay relatively high. They don't go down, unlike the cortical neurons. And the green cells, and the green is the EMG, which is the power that goes up and stays up. Okay? So the idea here was that you can think about this transformation maybe also as a neural integrator. Maybe also, like we have in eye movements, we have this integrator that we know about that was studied for the last 40 years. Also in the arm movement, we might need some kind of integrator downstream. And maybe in this case, again, there's an equivalent between cortical areas and spinal cord and cortical areas and brainstem. In the eye movement, it's cortex and brainstem. In the arm movement, it's cortex and spinal. Maybe the spinal cord network is also doing something like an integrator. Or between the spinal cord and the cortex, there could be an integrator. So that's, again, I just go back to the arm movement system to ask, well, is this relevant? And the answer is yes. You can have, this is still not proven. There's still a lot of work to do here. But this is the idea that maybe we can have similarity in the system, arm and eye movements in this relation. So now we can really summarize the whole, this part. But before, questions about this? Did this help? Was it too complicated? You can tell me for next year, for the student, for future students. You're helping future students. Was it too complicated? Okay. So to summarize, so we talked about we talked about all these things about motor cortex, and then I went to talk about dynamical system for three reasons. First, is an introduction for the talks for Thursday. Which will take the dynamical system approach and study the motor cortex. And then it was also an example for kind of transformation that the specific transformation that the brain can implement neural integrator. And again, it was an opportunity to show you a different system. We talked a lot about arm movements. It was an opportunity to show you that also the eye movement system and the kind of computation that we need to do in the eye movement system. In terms of preparing for tests here, level of intuition, that's what you need to know. I won't ask about specific connectivity matrices, how they will cause specific dynamics, but the general idea of that we can have neural integrators by tuning weights without the fine details. Also, the anatomy details of the eye movement are not really important. Just understand that we have this transformation from transient responses to sustained activity. And that's it. Any other questions? Good luck.
Thank you.
מידע עובר ממקום למקום. לא, באופן כללי, מידע עובר. מה החישוב שהוא עושה? איך הוא לוקח מידע אחד, הוא הופך אותו למידע אחר, מה הוא עושה בהם? כן. זה בטוח דעתה עליו. כן, כן, אני לא יודע. לא, 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 תשמחתי, יעלה עכשיו. אני חושב שאת ההרצאה הקודמת לא הדלקתי את המיקרופון. אי אפשר לראות אבל לא לשמוע. בחלק הראשון. לכן כדאי לבוא. אמרתי את זה פעם קודמת. מצטער, אבל לבשתי שזה דקות שמחתי להדליק את ה...
זה היה ביום חמישי. לא, כי הייתי ביום ראשון. אני לא. חמישי לא. אני לא חושבת שחייבים את החשבון הזה.
אני חושב שכן.
יש כאלה סטודנטים שירצו?
כן, באמת?
באמת? באמת?
הייתה